case study is in oncology, specifically focusing on cell cycle regulation. The core problem here is the inherent complexity of cell cycle networks and the challenge of establishing clear causality, understanding which events directly drive others. We leveraged high-resolution RNA sequencing time series data to capture dynamic changes. The goal was to reverse engineer the cell cycle to identify critical regulatory points. The application is directly relevant to drug discovery, helping us pinpoint the right target for therapeutic intervention. The diagram illustrates the different phases of the cell cycle: GON, S, G2, and M, and the genetic events that characterize each, which our models aim to decipher. To address this, we employed the cybernetic-inspired method, CIM framework, a multi-step approach for regulatory networks. It primarily uses transcriptomics time series data to understand gene expression changes over time. The framework is designed to identify key regulatory molecules and integrate diverse biological information, including pathways, genes, interactions, and modifications. Crucially, it captures temporal changes such as chromatin remodeling, which are vital for understanding dynamic biological processes. The workflow at the bottom of the slide visually outlines these steps, from unbiased gene selection to constructing a simplified network. The first step in our CIM analysis involved significant data reduction. From the initial high-resolution data, we identified 4,248 differentially expressed genes. We then narrowed our focus to a more manageable set of 86 canonical cell cycle genes and transcription factor genes. This focused approach allows us to concentrate on the most relevant players in cell cycle regulation, as highlighted in the network diagrams. Moving beyond simple correlations, our CIM framework uses information-theoretic methods to link genes with cause-effect relationships. This supports biologically realistic objectives by understanding regulatory logic, not just Q expression. Specifically, we applied time-delayed mutual information, TDMI. TDMI is critical because it helps capture hidden gene relationships beyond raw correlations, providing a more accurate picture of gene regulation. The diagram illustrates how these causal links form the basis of our network. The next stage of the CIM framework involves using a cybernetic ODE, ordinary differential equation, model to refine these transcriptomics networks. We introduced a variable G as a lumped regulator of gene expression, with activation or repression determined by TDMI. Our objective was to maximize the production of molecules essential for cell cycle function. We applied a generalized weighted maximization and incorporated change point detection to identify abrupt transitions in the time series data. A significant finding from this cybernetic fitting was the revelation of 3 distinct S-phase stages, which had not been previously identified, providing a deeper mechanistic understanding. This slide visually demonstrates the power of the CIM framework to capture complex biological dynamics. The multiple plots show the time series data, dots, alongside the stage-specific cybernetic model fits, lines. You can see how accurately our model tracks the changes in RNA fold expression for various genes across different time points, reinforcing its predictive capability. Here we see the results of our network refinement, which made the model both mechanistic and dynamic. On the left, we have the initial network with 86 nodes and 731 interactions. Through our cybernetic modeling, we refined this to a network, right, with the same 86 nodes, but significantly reduced to 153 interactions. The Kijai term defines these gene-gene interactions, with arrows indicating activation and dashes indicating repression. The line thickness is proportional to the strength of this interaction. Critically, validation showed that 90% of these interactions matched known protein interactions, demonstrating the biological relevance and accuracy of our refined network. Having achieved a mechanistic and dynamically validated network, our next step was to perform further network simplification. This aims to distill the core regulatory elements without losing critical information. For this simplification, we employed graph theory metrics, maximal clique centrality, MCC, and maximal RNA expression rate, MRER. 1,200 measures note importance by the size of the largest clique it belongs to. Cliques are sets of mutually connected nodes. The interpretation is that larger cliques represent more central and important nodes within the network. We successfully reduced the network from 86 nodes and 153 interactions to 66 nodes and 126 interactions, retaining only the most critical regulatory components while maintaining a 12 score of 7 or higher and MRER greater than 1, indicating strong functional relevance. The impact of this cell cycle regulation study is substantial. We successfully distilled a complex network into 126 critical links from over 700, making it far more interpretable. This led to discoveries of novel regulatory interactions, such as between c-Kone and BAT3. For validation, our model accurately predicted future cell cycles, a strong self-validation. The ultimate application is directly in drug discovery, enabling the identification of the right target with high confidence.